Okay, it is my pleasure to welcome you all to the annual SGP JGP Cranefield Awards Symposium. The winners, as you've seen on the slides here for 2023, who will be presenting their work today are Ivy Dick, Krishna Reddy, and Nicolle Godellas. And so we'll hear from all of them, and they will be introduced by Alessio Accardi, who's a member of the SGP Council and the chair of the Cranefield Awards Committee. So he'll introduce them in a little bit more detail, as well as introduce the Cranefield Awards themselves. So I think on the next slide, we have this outline of the schedule, and then which Alessio will review again for you as we begin. So I just want to say a couple of things about SGP before I introduce David Eisner, who will make a few comments about JGP, and followed by Alessio, who will be leading and moderating the symposium itself. So first thing I want to let everybody know is that all the planning and the organization for the 77th annual SGP meeting, which will be held in September of 2024 at Woods Hole, is on the topic of molecular evolution of ion channels and transporters. The date for that meeting is set. It's being organized by Lucy Forrest and Randy Stockbridge, and everything is well underway. You should be receiving information from Greg and from the organizers in the early part of 2024, and look for announcements to sign up to register for that meeting. So the second thing I want to remind everybody is that we have our annual business meeting coming up next week on Wednesday, the 20th, and I invite everyone to please come, and we'll give you updates on what happened at this year's annual meeting, next year's meeting, various awards, new initiatives that have begun this year at SGP, and we'll give you a little bit more information on the financial status of the society as well as plans for the future. And at that meeting, we will also welcome comments and suggestions from members for things that we could do better, for things that we might change, and for things that we can add. And that kind of dialogue has been very productive in the past. And also, I would like to invite everyone to, I think the next slide is the SGP JGP annual mixer, which the location hasn't been settled on yet, but it will be announced relatively soon. The time is Sunday the 11th, which is the usual time for this mixer. So I want to encourage everybody to look out for that and pay attention for your invitation, and please join us. And finally, I just want to comment again about becoming members of SGP. I think we have a really active, vital organization. We're really tuned in to thinking about efforts to promote the successes and the careers of young investigators, and we welcome comments and suggestions what more we can do and how we can do that better. So please, if you are a member, renew today, and if you're not, please go on the website and join us. So I'd like to turn over the mic quickly to David Eisner, who's the editor-in-chief of the Journal of General Physiology, to give you a couple of updates on things that are ongoing and planned.
Well, thanks very much, Jean. And certainly, I know the editors enjoyed very much reading the papers of the Cranefield winners, so I'm now looking forward very much to hearing them. So all I want to say about JGP now is to highlight some special issues we've got coming up. There's one which, as you can, has a deadline at the 1st of February 2024, which is in conjunction with the Mapping the Pain Landscape SGP meeting that was held at Woods Hole in September of this year. So that's the first of these. The next one on the next slide, we have a special issue on the subject of voltage-gated sodium channels with a deadline of August 2024. And last but not least, in conjunction with the 2024 Woods Hole meeting, which Jean has told you about, we're having a special issue on molecular evolution of ion channels and transporters. And I think that's really all I want to say today. Thank you.
Okay, great. Thanks, David. So now I would like to give the floor to Alessio Accardi, who is the chair of the Cranefield Awards Committee, excuse me, to introduce us to the awards and moderate the speakers. Alessio?
So thank you, Jean, and thank you, David, for giving me the floor. And it is my great pleasure and honor to introduce the Cranefield Awards this year. And these awards were instituted in 1996. The first award was instituted in 1996 in honor of Paul F. Cranefield, who was a very long-term head editor of the JGP for about 30 years. He was the head editor from 1966 to 1995. And obviously, the SGP and the JGP have a very strong, close relationship. And this has to do with the fact that there is a lot of scientific overlap between the interests of the journal and the society, and there is a lot of sharing of information and resources. And so when Dr. Cranefield, who was a professor at the Rockefeller University, retired from being the head editor of the Journal of General Physiology, the SGP instituted this award for a paper that was awarded to a young investigator who in the preceding year published an article of exceptional quality in the JGP. And the point, one of the important things to note is that in the establishing of this award, one of the points was that this should not be given every year necessarily. It was only if truly an outstanding paper came out in that year that it should be recognized. This is not to be given to anybody. This is just for people who do truly outstanding work. In 2005, when Dr. Anderson was the head editor of the JGP, there was the decision made to expand the scope of these awards, not only to include those to a junior investigator, but also to trainees. And so two more categories were added, and there were the student and the postdoctoral fellow Cranefield Award. And we have had many people who have received these awards have then gone on to have like stellar careers, in the sense that indicating that this is really a recognition of quality, both in young investigators and in trainees. And some have actually, and you can see the list here. And I think that the names, many of you will recognize many of the names that are here. Some are involved now still in the SGP, and some have been presidents and counselors. And then many of these have now gone, many of the trainee awardees have gone on to now get their own independent positions and their own independent labs. So this is really like a sort of like a gateway award in many ways. And so without further ado, we can go to this year's awardees. And the first talk is going to be presented by Ivy Dick, who is an associate professor in the University of Maryland School of Medicine. And she is in the physiology department, and she's going to tell us about the KB1.2 channelopathic mutations evoke diverse pathophysiological mechanisms. Ivy?
Thank you very much for that wonderful introduction. And I would like to thank as well the Society of General Physiologists and Journal of General Physiology for their support and for this opportunity to speak to you today. So today I'm going to be talking about the mechanisms underlying mutations in CAV1.2 and how they affect the channel. Sorry. There we go. So in 2004, Splowski and Keating uncovered the underlying cause of a severe form of long QT syndrome that caused a multi-system disorder, including craniofacial defects, syndactyly or webbing of the fingers and toes, neurodevelopmental delay or autism, and severe long QT syndrome, which was often fatal in early childhood. So the underlying cause of this disease was identified as a single point mutation within CAV 1.2. Since this first discovery, the number and variety of mutations within this channel has grown tremendously, with some mutations producing the full multisystem disorder, including the neurological deficits, while other mutations produce much more selective effects on just the heart, or some even just the brain. So then, my apologies, I seem to be missing a slide, but so today I'm going to be focusing on a number of these mutations, these 7 mutations right here, which actually seem to occur primarily within the distal S6 region, which I've highlighted in blue just for reference. And so to visualize the locus of these channels, we used a homology model of CAV 1.2 based on the CAV 1.1 structure described by Nan Yang. So what you can see is that, again, the S6 regions I've highlighted in blue for reference and pulled out here. And most of these mutations are sitting right at the distal S6 region, which is known to be important for channel trafficking. So to begin today, I'm going to start with the previous mutations that were discovered, sort of the canonical Timothy syndrome mutations, G402S and G406R, which sit within the 1S6 region of the channel. So to begin, we looked at voltage-dependent inactivation, which was actually the first thing looked at by Splowski and Keating in terms of how these mutations affect channel function. And you can see voltage inactivation, or VDI, as the decay in the barium current as it transverses the channel. And I've highlighted in blue just for reference. And what you can see is that both the canonical Timothy syndrome mutations significantly decrease voltage inactivation. They almost completely abolish it. If, however, we look now at calcium-dependent inactivation, we can see this as the stronger decay of the calcium current as compared to the barium current. And again, for reference, I shaded this now in pink. And what we found for the canonical Timothy syndrome mutations is both mutations significantly decrease this form of inactivation. So both forms of inactivation are decreased, resulting in somewhat of a gain-of-function effect. However, these mutations occur within the distal S6, which is known for channel activation. So we wanted to look at how these mutations affect the activation of the channel, so the open probability as a function of voltage here. And what we found is that for G402S, now we have a right shift in channel activation. Well, for the 406R, we had an opposite left shift in channel activation. And so what you can see is, even just looking at these very first 2 mutations, the mechanisms underlying the biophysical changes are actually starting to show a variety of different effects. And so we wanted to continue this. And to begin, we looked at these mutations, L762F, which was originally described by Jeffrey Kim, and E1496K, which, unlike the other mutations, does not sit in the distal S6, but does sit on the EF hand of the C tail of the channel. And this was originally described by Niels Decker's group. So again, we first looked at voltage-dependent inactivation, and now showing not just the exemplar current, but the population data as a function of voltage. Again, VDI is shaded in blue. And like the canonical mutations, VDI is almost completely abolished in one mutation with a more moderate effect in the other. However, if we now look at calcium-dependent inactivation for these mutations, so now I've highlighted it in pink for reference, what we find is that, unlike the canonical mutations, calcium-dependent inactivation is completely spared. So we're getting much more selective effect with these mutations. Finally, if we look at channel activation, again, the activation as a function of voltage, unlike the canonical mutations, we have actually no shift in channel activation. They look just like the wild type channel. And so now we're starting to get much more selective effects, in this case, on voltage-dependent inactivation alone. Finally, I wanted to look at these last 2 loci. We have 2 mutations at the I1166 loci, with the V mutation producing a cardiac selective effect, while the T mutation produces the much more severe, and one of the most severe forms, actually, of Timothy syndrome, including the neurological effects. And then I1475M in the S6 of the domain 4. Again, these sit right at the distal S6 region. And if we look at voltage-dependent inactivation, now, unlike all of the mutations that I've shown you, voltage-dependent inactivation is completely normal within all three of these mutations. However, if we now look at calcium-dependent inactivation, what we found is that to varying degrees, each of these mutations is decreasing the extent of calcium-dependent inactivation, or CDI. Looking at channel activation, what we found is that for the I1166V mutation, we get a small left shift in channel activation. However, it did not reach statistical significance. However, if we look at the T mutation at the same locus, now we get a very, very large left shift in channel activation, which is much more than statistically significant. This is a mutation that we've seen the most hyperpolarizing shift for any of the mutations. If, however, we look at this last I1475 mutation, despite the locus within the distal S6, there's really no shift in activation compared to the wild type channel. And so now we're actually getting a pretty diverse effect of these mutations, where some have selective effects on one form of inactivation and others have effects on differential effects on channel activation. And so given the importance of calcium-dependent regulation of these channels physiologically, we wanted to dive a little bit deeper into the mechanism underlying the deficits in CDI that I showed you. And so to do this, we first considered an allosteric model of channel gating. So within this model, channels initially open within this mode 1 set of states where they have a high open probability. However, upon calcium entry into the channels are driven into this mode calcium regime, which is characterized by a much lower open probability, causing that inactivation you see in whole cell experiments. Within this scheme, CDI has two major components. FCDI is the fraction of channels in mode calcium and is dependent on the calcium driving force, while CDI max is the maximum amount of CDI you would get if all channels entered mode calcium, and that's dependent on the open probability of the channel. And so what we realized is that FCDI can actually be bypassed so that you can directly measure CDI max if you simply provide the calcium at the mouth of the channel. And you can do this by calcium uncaging using a DM-nitrophin calcium cage. So we've done this for the canonical Timothy syndrome mutations, 402S and 406R. And what we did is we measured CDI as a function of that supplied calcium, and you can directly measure CDI max, which is about 0.75. So you see for wild type channels, there's a nice strong CDI. However, if we now do this for G402S, which has this right shift in channel activation, so these channels don't like to open, we find that if we actually replace the calcium ourselves, we get a nearly identical curve to wild type. So this mutation is a loss of FCDI. It's just a loss of the calcium driving force to reach mode calcium. If we do this now for 406R, which has that opposing left shift in channel activation, no matter how high we drive the calcium, CDI max is still significantly blunted compared to the other two channels. So now looking at this for these newer mutations, so just to explain the experiment a little bit more clearly, we started out at low calcium with this DM-nitrophin cage. Calcium is caged, so you have a nice low calcium measured by a fluorescent dye. Apply a UV flash, the calcium is released from the cage, and we can simultaneously measure the calcium concentration that we're applying to the channel. Now, we want our calcium source to be from the calcium cage and not from the calcium entering the channel itself. So we just minimize the amount of calcium coming through the channel by simply using a tail current protocol. So we only get this very brief spike of calcium entry. And that allows us to measure CDI as the difference before and after the UV flash. So when we do this for wild type channels, as I showed before, we get a nice curve with a strong CDI max. And then we wanted to apply this to our Timothy syndrome mutations. So looking at the I1166T first, this is the mutation that had that significant left shift in channel activation and actually the most severe phenotype in patients. And indeed, the CDI max is significantly blunted, just like what we saw for that G406R mutation, which also had that left shift in channel activation. If we now look at the 1166V mutation, which causes a slightly less severe phenotype and much less of a left shift in activation. We see the same thing, but again, that same pattern sort of appears where it's much less significant than the 1166T mutation, yet still a reduction in CDI max. And finally, we looked at I1475M. Now, this mutation had absolutely no change in channel activation, which makes it a lot harder to predict what we think might be happening here, whether it's a CDI max or FCDI change. And it turns out that this mutation, just like the other two, actually also has a significant reduction in CDI max. So all three of these newer CDI affecting mutations have a change in CDI max, which is a function of the open probability of the channel. And so that led us to want to do single channel recordings where we directly look at channel opening. So to do this, we apply a voltage ramp across a single calcium channel. And you can see the response here where the channels begin to open as the voltage increases. And they open to the level of the conductance shown here, which is dictated by the GHK equation as a function of voltage. So if we do this for many, many sweeps, we can define the open probability as a function of voltage. And we can also look at the open durations of the channel as a probability histogram. And what we see is, as expected for mode 1 behavior, you get these really rapid openings and closings shown as this sort of left short open duration histogram. Finally, we can actually change the opening of the channel by adding an agonist, BK. So this is known to cause the channels to shift into an alternate mode of gating where they open for really sustained periods of time. So you get this, it's called mode 2 behavior. And we do this actually mostly just to be able to count the number of channels within our patch and be sure that we actually know exactly what is there. So if we now do this for the I1166V mutation, what we find is that it looks, for the most part, a lot like wild type, but we periodically get these sweeps where we get this much longer opening. And this results in, again, a slight trend towards a left shift in channel activation, but it, again, did not quite reach statistical significance. However, it looks just like what the tail current showed. But if you now look at the open durations, you start to see that you're getting more of these longer duration openings. So this almost mode 2 behavior that flickers in and out, just like what we see with BK. And then BK has the added effect on these channels. We now look at I1166T. Now we start to see these longer mode 2 type openings occurring much, much more frequently. And as a result, we have a significant left shift in channel opening, as well as an increase in the open probability across all voltages. And so if we look at the open duration histogram, you can start to see that you get this larger population of openings that just have this really sustained channel openings. And finally, we looked at the I1475 mutation. If you look just at the exemplar data, what you see is these rapid openings. It looks a lot like wild type. In fact, the average data showed really no difference compared to wild type. Same thing with the open durations, and it still responds to BK. So this is interesting. We actually picked up no differences in the open probability, at least in barium, indicating that this mutation might actually be proceeding from a slightly different mechanism than some of the other mutations. It's interesting because this mutation is the only mutation in domain 4. Since calmodulin, the initiator of CDI, binds to that C tail, we wonder whether or not that could be a change in transduction to some effect. And so we have a variety of mechanisms leading to gating deficits of these channels. And we wondered whether or not some of these mechanisms and some of these effects could help explain why the phenotype in patients is actually somewhat different, where some patients have this severe neurodevelopmental delay or autism, while others do not, and they have mostly cardiac effects. So if we look at our data and compare it to the actual symptoms in patients, what you notice is that those mutations that have that really significant left shift in channel activation, those are the ones that also have the neurological phenotype, the really severe neurodevelopmental delay. And so we wondered if we could identify a difference in the response of these channels, even in HEK cells, to a neurological stimulus. So what we did is we applied a 40 hertz train of neuronal action potentials to the cell, and we recorded the calcium in response to that. And so here we're just plotting the amplitude of the calcium across this train. Now, if we do this for the I1166T mutation that has that really large left shift in channel activation, as well as severe neurological phenotype, you see a significant deviation from the wild type behavior. So the dashed line is showing wild type, and you can see that you're coming well below that line. Now, the only other mutation so far that I've described to you that also has that left shift in the autism phenotype is G406R, that original Timothy syndrome mutation. And what you see is exactly the same thing. It's deviating from the wild type behavior. However, all of the other mutations that have a long QT selective phenotype that only affect the heart look identical to wild type. So the wild type line is actually shown for all of these. There's just no difference with this behavior. Likewise, if we measure the duration of the calcium response to each action potential, so the half-width action potential response, only these two mutations that have that significant left shift actually have a deviation from the wild type behavior. And so what we think is happening here is that for the neurons, this activation, this enhanced activation, the left shift, and for the 1166, that enhanced open probability with mode 2 opening is really important for that severe neurodevelopmental delay phenotype. Now, that's not to say that VDI and CDI are not important in the brain, but in the case of Timothy syndrome, for that really severe phenotype, it does seem to correlate with activation. Whereas for the cardiac cells, it seems like inactivation actually primarily drives the long QT phenotype with a contribution of activation. So far, everything I've shown you has been in HEX cells, and I wanted to spend just the last couple minutes of my time and show you some preliminary data as to where this is going and how we can actually look at these mutations in a somewhat more native system. And so to do this, we utilize induced pluripotent stem cells, and to begin with, we differentiate them into neurons. And so you can see the neurons nicely here. When we do this with wild type or G406R mutant cells, which were actually a kind gift from Masa Yazawa over at Columbia University, we find that inactivation is again decreased as seen in the HEX cells. So it shows that they're a nice model system for this. And when we look at the action potentials of these cells, the electrical properties, what we found is that the cells actually have two functional populations, those that respond to a stimulus with a repeated firing, and those that no matter how large our current stimulus is, we only get one action potential. And this occurs within the Timothy syndrome cells as well. But what we found is that the proportion of neurons that fire repeatedly versus single firing is quite shifted in the Timothy syndrome cells. Moreover, if you look at the frequency of firing within these repetitive firing cells, the TS cells seem to fire with a much faster frequency. So we do have a change in the electrical properties of neurons that are expressing the G406R mutation. And then finally, what we wanted to look at is cardiomyocytes, since these mutations all cause effects in the heart. So again, we looked just to make sure that the mutations do recapitulate what we see in the HEX cells. They decrease the inactivation of the channel. And now if we look at the action potentials of these cells, what we see is for G406R, we get these really, really fat action potentials that are indicative of the long QT phenotype. Same thing for the more selective mutations. We get these prolonged action potentials, although to a lesser degree, even in the mutation that has a single loss of CDI, although I will say that the ends on this are actually quite low. So this is a work in progress. But looking at how the arrhythmias actually can occur in these cells, we can watch the action potentials as they propagate across the monolayer of these cells. So this is a voltage-sensitive dye recording, and you can see that the action potentials propagate smoothly for wild-type cells. However, for the G406R channels, they routinely enter this sort of spiral wave pattern, which is indicative of this arrhythmogenic behavior. if we do this for the other mutations, so we see exactly the same type of thing, the only difference is the propensity to actually enter this arrhythmogenic pattern. So you get much more entry for these 406R and 116016 mutation, well, for I1475M often looks smooth, and then as you pace it, it will start to enter these spiral patterns. And so hopefully I've convinced you that mutations in CAV 1.2, they can actually have quite a variety of effects on channel gating, with some having very specific effects on either inactivation or activation. And that can somewhat explain, at least partially, the difference in the phenotype of these patients. And so with that, I would like to thank the members of my lab. So Maura Bongoy, who was the lead author on this paper and did a lot of really wonderful work in HEX cells. As I'm a little bit over time, I don't have time to name everybody, but all of the members of my lab are moving forward with those IPS cells, which is not a trivial thing to do. And I'm really appreciative of all the work that they've been doing. Masa Yazawa gave us the initial patient-derived Timothy syndrome cells. And now Debbie and Emma have been CRISPRing new mutations into these cells. And of course, thank you to our funding agencies, and a special thank you to SGP and JGP for giving me this opportunity. And with that, I will take any questions.
Thank you, Ivy. This was a fantastic talk, and congratulations again. Questions? I see already one hand raised. Amy?
Yeah, congratulations, Ivy. This was a really beautiful talk. Very cool story. It was very interesting how you were able to biophysically dissociate the effects on the channel and then correlate them with either the nervous system or cardiovascular dysfunction. I just am recalling work from Ricardo Dolmetsch about these potentially non-calcium dependent effects of some of the Timothy syndrome mutations. And given that the left shifted mutations really cause this huge gain of function through a voltage dependent mechanism, I'm just wondering, do you think it's the calcium or maybe kind of voltage dependent other stuff that is driving the system to some of these defects?
So that was actually one of the reasons that we did these first experiments in HEX cells. So HEX cells don't have any of the downstream signaling, any of the neurological proteins that might also be affected by changes in scaffolding, changes in the RAS pathway that Dolmetsch talked about. And so these effects that we're seeing can only be caused by changes in channel gating. Whether or not that's calcium specifically, because calcium driving the inactivation deficits could be different, or a conformational change due to that voltage sensor moving, like what Dick Chen showed. Both of those would be at play here. They're both consistent with our results. And honestly, it's probably a combination of both. But this does indicate that the actual gating of the channel and the requirement of the channel to move is important in the downstream process, in the effects of these mutations. Not to say that other developmental effects aren't also at play, but we do think gating matters.
Thanks.
Okay. Any questions? Oh, two more questions. Jean?
I think I have a million questions, actually. This was a fantastic talk. I really like this story. So I wonder about several things. One is splice variants of CAV 1.2. So I think CAV 1.2 has a lot of different splice variants that are different in different tissues. And so I guess you're not dealing with that here, or maybe you are, and maybe you don't know so just what the role, and then also, do Timothy syndrome patients have a vascular phenotype also?
Yeah, so for splice variation, I just couldn't fit it in here. We do have some other work that's targeting that. So the mutations themselves, for it's going to be way at the top, but for the canonical mutations in that 1S6 region, right here, those are going to be within a mutually exclusive exon, either exon 8 or 8A. It turns out that the cardiac expression and the neurological expression of those exons is very similar. And in fact, the lower expression of the G406R mutation because of that splice variation is probably why you see more of those patients surviving. So if you look at the number of patients, a lot of these mutations only have 1 or 2 patients. G406R it was 25, it's higher now. I think it's 27 or 28. But there's a lot more mutations occurring within that mutually exclusive exon because you don't have as much expression in the heart, I think. And I do think that that's something to be dealt with. The actual functional impact of the mutations. So we've directly tested it for the canonical mutations. If you do 8 or 8A in the different splice, the 2 major splice backgrounds, biophysically it looks the same. The impact is the same in either background. We haven't extensively done the other splice variants yet. And then, sorry, what was the second question?
I just wanted to know whether the individuals have vascular phenotypes.
Yeah, they do.
Okay.
So there's actually a lot more phenotypes being recognized than what was even on that slide. So it is affecting blood pressure to some extent. It's just that a lot of the described phenotypes that the doctors focus on are the ones that, you know, are potentially lethal. So they get a lot more focus. But when you talk to the parents of these patients, the phenotype, there's sleep disorders, there's all sorts of things happening beyond just the cardiac and neuronal phenotypes that I sort of emphasized today.
Great.
So everywhere there's a calcium channel.
It's great. Thank you. Thank you.
Great. David?
Nice talk. I'm always impressed by how much faster calcium-dependent inactivation is than voltage-dependent inactivation in a ventricular myocyte. So if you have a pure mutation that only affects voltage-dependent inactivation, is it surprising that this produces effects on the heart, arrhythmias?
So in the hex cells, we manipulated the experiments so that in the CDI experiments, we have a lot less VDI. In the heart, you have different beta cells there, so you have a lot more inactivation. There we go. You have a lot more inactivation, both VDI and CDI in these heart cells. This is actually calcium, so both VDI and CDI are at play, and which is why we get a much larger effect of the 406R, especially towards the end, because that VDI deficit in addition to the CDI deficit. So the mix of beta subunits does increase the effect of voltage-dependent inactivation in a heart cell. But we actually think that faster and that stronger calcium-dependent inactivation is a little bit more critical, which is why even the mutations that have very selective, I guess it's the next slide, that have very selective effects on calcium-dependent inactivation still have significant effects in the heart, even though it's actually a very moderate biophysical effect when it's expressed heterozygously with the beta subunits where you have the VDI intact.
Okay, John?
Wonderful. Hi, Ivy. Just thank you very much for doing this. My daughter's close friend is an LQT8 sufferer, and I've been and your hypothesis I think is can profoundly help a lot of people understand what's going on with their loved ones. And my question for you is with this slide here exactly is, do the changes in action potentials you see are cardiac ones are quite extreme. Do those correlate well with the QT interval from the patients that have these mutations? Are they more or less severe in a dish?
They're more severe in a dish. And actually, I've always waited for someone to ask me that question. I'm always surprised no one does. So thank you. So first of all, these are at room temperature, at least for the patch clamp recordings. So you do get some prolongation. But we actually find that, and this is true for any of the long QT mutations we've studied for calmodulopathies as well, we get more severe phenotypes in the dish than you get in the patient, because this right here would be lethal. You would never see a patient have that. So I think what it is that in the dish, the cells can survive this, but in a heart, the entire heart cannot. You also notice that when we put them in the monolayer, it gets a little bit less severe. So when you have a syncytium of cells all acting together, you get less of these sort of outlying, really, really prolonged action potentials. So when acting together, they do seem to correlate a little bit more with patient phenotype, but we always see something a bit more severe than in the patients. And that's across the board with the iPSC cardiomyocytes and long QT mutations.
Thank you very much.
Okay, any other questions from our attendees, any of the trainees? If not, then I think we can thank Ivy for a fantastic talk again. This was really exciting. And then we can get on to the next speaker, who is Krishna Reddy, and who is a postdoctoral fellow in Olga Budkar's lab at Weill Cornell. And he has been working on an archaeal glutamate transporter. And he's going to, do we have the title, Greg? Okay, perfect. He's going to tell us about the heterogeneity of substrate binding in glutamate transporters.
Great. Thank you so much, Alessio. Hopefully everybody can hear me and see me and see my slide. Yeah, thank you so much to SGP and JGP and the Cranefield Awards committee for this incredible honor. And what I'm going to talk to you today about is my manuscript in JGP in 2022. Hold on, if I can figure out my laser pointer here. There we go. You know, which is about the heterogeneous substrate binding in a glutamate transporter homolog. And I struggled for a while to think about how to present this paper, because if you read the paper, it's all very nicely and logically laid out. We made this interesting observation about heterogeneous substrate binding. We did some mechanistic and structural studies to characterize this, and we contextualize this to how we think this affects transport. And it's all very clearly and I think nicely laid out, but this is not at all my relationship with this paper because, you know, like I think a lot of science is not a really straightforward and logical endeavor. There's a lot of twists and turns and unexpected things. And really my relationship with this paper is much more like this, where we started with a completely different question and then stumbled on this interesting finding and then worked really hard to contextualize this and understand what was going on. And so hopefully what I can communicate to you today is not only, you know, why I find this finding really interesting and important to how we think about ion-coupled transport, but also, you know, personally and professionally, how important this paper was to me as a scientist. So our lab I'm in Olga Budkar's lab, and we're really interested in how glutamate homeostasis works at synapses. And so basically how this operates is at a presynaptic neuron, you have these membrane vesicles, which are packaged with the neurotransmitter glutamate. And upon a signal, this fuses with the presynaptic membrane, releases glutamate to the synaptic cleft, and then this hits glutamate-gated receptors on the postsynaptic membrane. And this is one of the key bases of glutamatergic signaling. But there's also kind of a energetic challenge that happens here. So once this glutamate is released into this cleft, it reaches maybe up to a micromolar glutamate concentration. But it has to very quickly escape this region, otherwise it'll continue hitting these glutamate receptors, and that can lead to really devastating consequences for the neuron: excitotoxicity, cell death, and this is associated with a lot of neurological disorders. And so the way that nature has solved this energetic challenge is with glutamate transporters. And it needs transporters because it wants to recycle this glutamate. It has to go back inside the cell, but inside the cell, there's very high concentrations of glutamate, you know, several fold above what's in the synaptic cleft. And so this is going against its concentration gradient. And transporters are able to solve this challenge using energy from ion gradients. And these are called the excitatory amino acid transporters, and they use sodium, potassium, and protons to drive transport of glutamate against its concentration gradient back into glial cells. And this allows for another round of glutamatergic signaling. And so in our lab, we're very interested in these glutamate transporters, what they look like, how they work. And again, this is showing the mammalian excitatory amino acid transporter called EATS. And they, as I mentioned, they use sodium and protons, and also the counter transporter potassium. But much of our knowledge of these transporters come from simple prokaryotic homologs. And what our lab has spent a lot of time characterizing is a prokaryotic homolog found in deep sea thermal vents called in an archaea. And this transporter is called GLTPH. And GLTPH is a little simpler. Instead of using 3 different ions, it just uses one ion. It just uses one type of ion. It just uses sodium, and it couples a symport of sodium to the symport of the substrate. And so a lot of our understanding of the mammalian transporters comes from this prokaryotic transporter, because the sodium sites seem to be conserved and the coupling mechanism seems to be quite consistent. And so what I'm going to do is slowly walk you through what we know of sodium coupling, and kind of the first way I do this, kind of inspired by Olga, is anthropomorphizing it as an elevator. And then we'll kind of go into what we think about and how this kind of connects to protein structure. And so the key to ion-coupled transport in these transporters is that if you imagine a transporter like an elevator, the aspartate, this green guy here, is unable to enter the binding site because it's occluded in some way. And so what it has to do is be able to move this occlusion in some way, and it does this with the help of sodium. And so sodium is able to remove this occlusion and expose this binding site for aspartate. Now, an important part of this transport cycle is that sodium alone cannot go through and move this elevator. And this is kind of key to tight coupling. If the sodium were able to just go back and forth, that would be a sodium leak, and that would just kind of be devastating consequence for the cell. And so instead, now aspartate and one more sodium is able to come in, and now they have this key to be able to close the gate. And now this is kind of the basis of ion-coupled transporter in this family of transporters. And so this is kind of the anthropomorphized version of this. And so now I'm going to this is basically the same thing, but with protein structure. And now, before I start this movie, what we have is a very rigid scaffold domain, or trimerization domain, that's in wheat here. That doesn't really move all that much throughout this transport cycle. And then you have the mobile transport domain, and this is in blue here. And this gate here, which we call HP2, but this is where everything kind of binds in this little crevice here. And this is kind of built on several structural and mechanistic studies over, the course of about 20 years at this point. Okay, and in purple are these sodium ions, and in green here is our substrate. So I'll start the movie now. So as I showed you with the elevator, the sodiums are able to enter first. This exposes the substrate binding site, and now aspartate and this last sodium is able to come in. And now, much like an elevator, it's able to go to this massive conformational transition from what we call outward-facing state to inward-facing state. And so it's able to traverse this membrane in this giant rigid body movement across the membrane. And once it gets to this inward-facing state, the solutes can release first the aspartate and then the rest of the sodium ions. And then spontaneously, this transport domain that's empty at this point is able to come back to the outward-facing state and is now reset for another round of glutamate cycling. And the question that's always kind of asked about these transporters is, do they, are they kind of monomeric? Do they occur in isolation? Usually I show like a trimerization structure, and because these do operate as trimers. But very recently, a student who previously rotated in the lab defended his PhD, and he gave Olga this really beautiful creative rendition of the glutamate transporter that shows its kind of trimeric stoichiometry. And what I especially love about this picture is it's actually scientifically accurate in addition to beautiful. So if you notice, these flowers here will correspond to the transport domain, and these leaves here are your scaffold domain, and we can kind of correlate it to this here, right? And if you notice, what Chang Hao did was he flipped one of the flowers in this little art piece here. And I think this makes a really important scientific point, which is that these protomers can operate independently. They're not coupled to each other in any way. And I think this kind of nicely shows the asymmetry of these transporters. And so, over the course of 20 years, this transport has been really well characterized in this kind of simple transport cycle of ion-coupled substrate binding. But, in recent years, there's kind of been a wealth of literature that, as the techniques get more advanced to study these transporters, we find more complexity in them. And so what our lab and many other labs have now begun to do is look at individual transport molecules and look at their kinetics and how they transport across the membrane. And so this is just an example of kind of a single molecule FRET setup where we can basically put a donor and an acceptor dye on two side-by-side transport domains and map out if the transporters are either in outward and if the pairs of transporters are either in the outward and inward state or the outward and outward state. And so in this way, we can get an idea of conformational dynamics of this transporter. And, what all these papers, and there's probably more I'm forgetting, have started to paint a picture of is that these transporters just don't go into this homogeneous transport cycle. There's some underlying heterogeneity that structures can't capture. And so this is kind of an example of a single molecule FRET trace, again, where we're mapping conformational transitions, but this isn't unlike an ion conductance looking at open probabilities. It's kind of the same conceptually. But basically what you could tell from this individual trace here is that for a very long time, it'll be stuck in one type of conformation. But then and this is kind of this non-dynamic period, but then after several seconds to minutes, it'll start going in these rapid up and down conformational transitions, and then it'll again pause for a while, not really doing a whole lot, and again up and down, very dynamic behavior. And so this is what we call kinetic heterogeneity or modal heterogeneity, basically that individual transporters can kind of go up and down or pause and have these kind of heterogeneous transport activities. And so the phenomenon has been very well observed, but structurally, nothing in all these fancy cryo-EM structures or crystallography over the years had really never given any clear insight as to why that might happen. So we have this discrepancy between what we see in structure and what we see in the kinetics of the transporter. And so this is the question we ended up answering, I think, but this is not where we started, as I mentioned. So we started with a completely different question, and I'm not going to belabor the point here, but basically we had this engineering challenge, right? We were basically saying, okay, we have these sodium coupled transporters. Can we make them into proton coupled transporters, right? And, of course, we ended up answering a completely different question, which is what's the deal with weird substrate binding, and can this explain kinetic heterogeneity? Almost Seinfeld-esque. And so the way we started this project was we aligned the known sodium-coupled transporters and proton-coupled transporters, and just when I was entering the lab. And the idea was we looked at conserved sites and we're basically saying what could explain this ion coupling difference. And there were two mutations that jumped out to us. We tested many more, but there's only one we truly characterized, the serine to glutamate here, and this aspartate to asparagine here. And the reason these mutations were interesting to us to begin with was if you look at the structure, these surround sodium 1. And so the kind of very naive idea that we had was, okay, if we mutate this aspartate here, we're going to abolish sodium binding. And if we mutate this residue here to a glutamate, we're going to introduce proton binding. And this is going to magically change the transporter. And, when I joined the lab, the postdoc who was doing this project had just left. And Olga was like, oh, okay, we'll just get a few figures, do some characterization, and it's a quick paper. PIs never say that it's going to be a quick paper, no matter how much you believe this. That's my unsolicited advice here. And so I think, but what came out of this study, I think, was a failure so spectacular that it ended up turning out pretty good. And so I like this kind of meme here, that the task did fail successfully because we ended up stumbling on something completely different, but still, equally interesting. And so this was the experiment that kind of started us all off down this road. And this experiment is called isothermal titration calorimetry, or ITC. And so the basis of this experiment is that in this sample cell here, you have your apoprotein, and you're slowly injecting ligand over time. And basically what happens is you have it compared to a reference cell. This is water. And the idea is as you inject your ligand, it causes some binding reaction, which either releases or absorbs heat. And then as it does that, there's some constant power that's applied. And basically, you're trying to, the instrument is trying to supply power to maintain a constant temperature. And the idea is that by kind of integrating all this information about how the power is distributed over time, we can get a complete set of thermodynamic parameters of substrate binding as you go from apostate here to the completely bound state here. And so we can get enthalpy, entropy, we can get the stoichiometry of the reaction, and of course we can get affinity of this interaction. And so what you'll notice from this figure here is that for a very simple one-site binding mechanism, the curve is nice and sigmoidal. And this is kind of very typical of a ITC one-to-one binding experiment. Okay, and then we did that with, so one more point here. And so the experiment we want to do with our protein is, and with sodium coupled transporters in general, is as you increase the sodium concentration, you're also increasing the affinity for aspartate. And so with these kind of principles in mind, we went about this study with this mutant transporter here. And this does not form a sigmoidal binding curve at all. And it forms this really funky bimodal curve. And it took us, many months to even convince us that this was real. And so what you basically see here is you kind of have this, initial few injections, and then kind of close to saturation, you see, this greater enthalpy kind of peak here. And then finally, this is what saturated protein looks like here. And an important point about this experiment is that we did this in really, really high salt concentrations, saturating what we think is saturating sodium concentrations. So we don't think that we're not completely occupying the sodium sites. And we have some other evidence also kind of laid out for this. And so why does substrate binding look so weird? So we kind of did a lot of literature reading and trying to make sense of this. And what we eventually came to the conclusion of is that these are two slowly exchanging non-identical binding states. And so the way you can interpret a curve like this is that with these initial few injections, what's getting populated is a higher affinity but lower exothermic enthalpy site. And then as you kind of go along titrating your protein with ligand, you now begin to populate a lower affinity but a higher exothermic enthalpy binding site. And so, and the reason why we think this is slowly exchanging, for many reasons, but one of the reasons is the time course of this experiment is between each injection is several minutes, and the experiment takes many hours to complete. And so we think that these are in a very slow equilibrium with one another. And we have some other experiments in the paper to kind of justify this. And so what exactly is going on with these? Like how do we actually have any idea of why this is happening? So one of our key observations, so as I said, we're in saturating sodium concentrations, but another way to confirm this, if it's a salt-dependent effect or not, is by basically taking the same sodium concentration, but now just changing the counter anion in the salt. And so in this case, and we can kind of extrapolate the population of the second site. And so we calculate the second site in sodium chloride conditions to be 26%. And we can modulate this just by changing the counter anion. And so we can do this in gluconate. And this is a little bit harder to measure. So this 22% value is not nearly as robustly calculated, but we can clearly see that this is a smaller peak here. And then we take another anion, in this case nitrate, and now we can see a much bigger peak, and we calculate the population of that second lower affinity site to be much higher. And so from this, we can see that the relative populations of this low affinity site are regulated in some way by the salt's identity. So now we're kind of in this conundrum of how and why does our GLTPH have two substrate binding states? And so, for the course of a couple years, my task was basically to systematically go through the possibilities and eliminate them. And so the first possibility, which Olga suggested, was that perhaps I miscloned the protein, or perhaps I somehow transformed two different constructs at the same time. I know Olga's listening. I'm still mildly insulted by this. So no, it was not because I miscloned the protein. The next possibility is that somehow we introduced some artifact. Maybe our mutations have changed something in some dramatic way. And we did systematically go about and figure this out, right? So we we figured out that we pretty conclusively stated that the mutant itself is not the problem here. So we still only observe one substrate binding site. We're not spontaneously making some other weird site away from the protein. And we also observe it in wild type, and not quite in the same way that I showed you in that bimodal thing, but it's still quite dramatic. So in wild type GLTPH, this is what it looks like. So we're comparing just two different salts, and you could see here in gluconate salt, which is here, you have a completely different profile versus when you just swap the anion. And this is in nitrate here. And so this, I think, was pretty dramatic for us to see that wild type has the same properties of heterogeneous substrate binding. And it doesn't seem to bind to other conformations, and so at this point we were kind of stuck what's going on. And then we started thinking about the anions, and so we knew that anions were potentially changing function, and or that they were changing functions. And the two ways you can think about this is the transporter, as it undergoes this conformational change, we know it has some uncoupled chloride conductance. It basically, as it undergoes this elevator mechanism, it spontaneously forms this pore that is able to pass chloride. And at this time, there had not been a structure of the chloride conducting state. The other aspect of anions is that they could just be generally causing some effects on proteins, whether it's folding or salting out or solubility. And coincidentally, the trends that we saw of increasing site 2 with different anions, they have that same kind of pattern in both of these different effects. So we were really unclear what was going on. So we turned to cryo-EM to try and figure this out. And the way we did this was we basically optimized our parameters to look at this second lower affinity state. And we collected data in two different ways. One, that substrate was present throughout the entire purification. And then the other, which is before equilibrium is formed, we added substrate a few seconds before freezing. And what ITC suggested is that the lower affinity state should be here in the before equilibrium data collection, but not here in the equilibrium data collection. And so we got some really beautiful structures. Actually, this is still my highest resolution structure, and this is the first data collection I ever did. So it just goes to show a lot of this is dumb luck, I feel. But we were able to see a lot of really beautiful side chains, 2.2 angstroms, but who's counting? And but most importantly, it's entirely in the outward facing state. There's no justification for a chloride conducting pore to be happening through here. And so we can pretty conclusively eliminate that it has anything to do with chloride conductance. And so we, at this point, we thought that it may be due to just general chaotropic effects of the protein. And, you know, at this time, the pandemic hit. So we really were stuck at like, okay, what is going on? We weren't able to do experiments. And so at this point, I just kind of threw myself in the data processing, frankly, because I had nothing better to do. And after many months of trying to sort through data and trying to understand and, you know, explain a lot of these things that I've already mentioned to you, we came to one hypothesis, which we think is quite convincing. And so what we saw is in the pre-equilibrated set where substrate was added just before freezing. Again, this is where we expect to see our binding heterogeneity. We're looking at the top of the transport domain. And so this is basically looking at the extracellular helices. And we saw this interesting mobility here. We were able to refine several classes that just had these different helical tilts that were going on. And these are quite good structures that we're able to refine. And so we saw that there's this kind of weird mobility of the extracellular helices, but this is only found before equilibration of the protein. When protein is purified with substrate around the whole time, we're unable to see this phenomenon. And so we tend to think that this is a nice kind of cohesive explanation of all of our findings here. And what another interesting observation, which kind of contextualizes that whole thing, is we saw in our very high resolution structures, a lot of structured waters around the ion binding sites and the substrate binding site. And what we found really striking was that it was only found in this lower half of the protein. You basically cut the transport domain in half here and see that all of these kind of hydrated regions are on the bottom half of the transporter. And so the way we think about this is that you have this more hydrophilic half, which is critical to ion binding, and that's strongly stabilized by polar interactions like water and ions and substrate. And then you have this extracellular half, which is very hydrophobic. And that has these weird dynamic properties that can be modulated by these chaotropic anions, such as substrate binding affinity and rate. And so, you know, with my last minute or two, I'm going to try and see if, you know, so did we actually come to a conclusion? Is this a structural basis of long-lasting kinetic heterogeneity? And we still need some convincing to do on it for ourselves, but we tend to think this is a good hypothesis. So kind of in parallel in the lab, Gerard had figured out some gain of function mutations of the transporter, which had different transport rates that affected this kinetic heterogeneity. And most of them were found in this very region, in these extracellular helices. So these are 3 of the mutations he characterized. And we were able to actually go back to our original question that we're asking is, can we switch uncoupling preference? And this ended up being relevant there too. And so this is a preprint that just came out. Again, we were trying to answer this question of how can we convert sodium to proton coupling? And we found in between an intermediate ancestral transporter that had broken ion coupling. So it's able to bind substrate without any ions. And what we found is that if you look at GLTPH, upon sodium binding, there's these massive changes in helicity. But when we looked at our uncoupled ancestor that's unable to couple substrate binding to ions, there's really not a lot of changes that happen. And so we think that even this extracellular helix mobility and packing seems to be a critical component of ion coupling in these transporters. And so to conclude, we very serendipitously found that GLTPH binds substrate much more heterogeneously than we thought. And before equilibrium, these helices are kind of variably packed, and this maybe sets the affinity of the transporter to be heterogeneous. And we propose that this could be a plausible basis of this weird kinetic heterogeneity that we experience in transporters. But as I said, this kind of journey was messy for me. And I think a couple other things that I found is that research often leads you in unexpected directions. And part of our duty as scientists is to be able to manage this and also follow the directions when they're interesting and might lead to cool places, but also know when to quit and or also when to move on. And I think in my specific case, failure can often be the first step in the direction of success. And I think this was, I kind of think back on this whenever having a tough time in the lab. And then finally, if your PI suggests an idea for a quick paper, you should run like hell. And so with that, thank you for your time. I want to thank, jokes aside, Olga was instrumental in guiding me along this journey to, making this from a weird observation into what I think is a really nice paper. And then Didar and Amanda were kind of critical in, some of the data collection of this. And then, I had a lot of help with EM processing and core facilities. And I would especially like to thank SGP and JGP for their support and this honor of this award. So with that, I'll take any questions. Thank you.
Thank you, Krishna. This was a very exciting talk. And not only the science, but also the life lessons. Thank you very much. And now the floor is open for questions. Well, let me start by breaking the ice. First of all, the changes that you're seeing in both the affinity and the directionality of the heat that is being released are quite dramatic.
Mm-hmm.
Do you think that the subtle movements, the subtle rearrangements and the differential water occupancy is sufficient to account for these very large thermodynamic changes that you see in the ion binding? Is there a way to quantify that and look at that a little bit more in a sort of rigorous,
were you one of our reviewers?
No, I cannot. I cannot.
No, we racked our brain on how we can directly show this, and I still don't know if we have a great answer on how we can directly show this. But yeah, it's a great point, right? Can we directly connect these helical movements on the time scales we're looking at with this kind of kinetic heterogeneity? So one thing I will say that I didn't show the data of is we can look at transport, right? And we can see that all we have to do is replace the anion, and we do see changing transport rates. But, as far as directly connecting the structural, observation with the timescales of ITC and all these observations, it's difficult. And that's something we're still thinking about.
Okay. Any other questions? Oh, I see a question. Jean?
I have a really naive question. So you have this transporter, and I get the idea that it changes conformation structure and basically flips. But then why do the ions why do they leave? Why do they unbind at that point? What happens? Is
Oh,
it a
or we're just
conformational?
talking about the general transport cycle. Yeah, so
Yeah.
it's a great question, and I didn't really make the point particularly poignant. So in this kind of model archaeal transporter on the inward face in the cytosolic side, first of all, the concentration of the solutes are much lower, and then second of all, the affinity of the sites are lower. So those things together are what allowed for rapid release of these things.
So the affinity actually changes when I see, okay, interesting. And I heard somebody say the other day that luck is an attitude.
That's great, yeah.
Hello. I was wondering if you could explain how the heterogeneity changed the the heterogeneous passage of ions was dependent on the monomer or multimer structure. I don't know. You might have
Yeah, no, it's and perhaps I wasn't clear. First of all, I know you're not Rada. I know Rada. That's not Rada.
Yeah, no, I'm Tristan. I got forwarded the link. I don't know where I'm coming from, but yeah.
No, fair enough. So actually, and perhaps I didn't make this point clear, is that we really, and actually, we were wondering for a while, was that actually the case, right? Like, is there something with the trimerization and the oligomerization that was affecting things? And there's kind of a large body of literature in the field and everything suggests that they function completely independently. And so we don't think that it has anything to do with the oligomerization status of the trimer, or that there's inter-subunit effects of ions. We think it's completely constrained within a single subunit.
Oh, okay, cool. Thanks.
David?
I'm afraid it's another ignorant question. Does the mammalian transporter require the other cations beyond sodium just to be able to pump up a bigger gradient? Is it?
I think so. So basically, and I think the other aspect of it is it kind of adds some additional brakes onto the transporter. And so you have, so the rate limiting step in the archaeal homolog, GLTPH, is a translocation from outward to inward facing state and then release on the inward facing side. In the mammalian transporter, it's actually potassium coupled antiport. And so that's another aspect of the whole thing, is that it seems to just add on a couple extra brakes and some complexity onto the transport cycle.
Okay. Thank you.
If we have no more questions, then I think we can thank Krishna for a great talk.
Thank you.
And thank you very much. And now we are on to to Nicolle Godellas, who is the awardee for the student category, and she won it for a paper entitled Probing Function in Ligand-Gated Ion Channels Without Measuring Ion Transport, which is the dream of biochemists in large part. And Nicolle was a grad student in the Grossman lab, and she just successfully defended. So congratulations, Nicolle, and now take it out.
Thank you, Alessio, and thank you to the SGP and JGP and the Cranefield Award Committee. I'm excited to talk to you today about actually my first project with Claudio, where we work to implement an alternative methodology to probe function in ligand-gated ion channels when measuring ion transport was not an option. So, okay. So I thought I'd talk about a little bit of a timeline first to get a better understanding of why we needed to implement this alternative methodology. So as you can probably tell from my title, we are a lab that studies ligand-gated ion channels, and we typically do so by means of physiology, which I know all of you are quite familiar with. And so many years back, probably when I was just starting, a little bit before I just started as a graduate student, we were working on a project that focused on studying the interdomain region of pentameric ligand-gated ion channels. And along the way, we discovered some mutants that had an electrically silent phenotype. So these particular mutants expressed really well on the plasma membrane, but when probed electrophysiologically, they appeared to be non-functional or dead. So we knew that we needed a new method to evaluate ion channel function and gain mechanistic insight into these particular mutants. So we decided to work to implement ligand binding assays, probing from the perspective of the binding sites instead of from the perspective of the pore domain. And Claudio and I quickly came to realize that ligand binding assays were helpful for studying more than just electrically silent mutants. So say you have a channel that desensitizes too quickly and you can't measure any currents, or for example, if you want to work to modify the lipid composition of your plasma membrane, say you want to deplete the membrane of cholesterol, and this might make forming those high resistance seals a lot more difficult. Along similar lines, say you want to study how your receptor functions in different kinds of membrane mimetics, so how it functions in a nanodisk versus a more native type of environment. So these are just a few examples of when ligand binding assays could be a great alternative to electrophysiological techniques. So we decided to perform competition ligand binding assays, and there are essentially two types of these kind of assays. So you have your equilibrium type assays, so you have your receptor of interest, your labeled and unlabeled ligand, and you mix everything together and you let it incubate until equilibrium, or as close as you can get to equilibrium, is reached. And then you have your protection type assays. So these are a little bit different. So you first incubate your receptor and your unlabeled ligand, which tends to be your ligand of interest, and you allow those two reaction components to reach equilibrium. And only then do you add your labeled ligand to the mixture. And the idea is that your labeled ligand binds to sites that are unoccupied by your unlabeled ligand. So in this case, our labeled ligand would be acting as a mere probe. So Claudio and I decided to start to implement equilibrium type ligand binding assays. So this is the direction that we went. And these are classical type of assays. So we had this kind of initial inclination to, okay, let's go to the literature, let's find a good protocol, let's get started. But we quickly came to realize that there were a lot of inconsistencies in protocols. So reaction mixtures were being incubated anywhere from 4 to 37 degrees for tens of minutes to tens of hours. And we also started to notice that the lines between protocols of equilibrium type assays and these protection type assays started to blur. So things were a little bit complicated. So we wanted to go ahead on our own and work to optimize this methodology for our purposes. So for our optimization, we decided to use the homomeric human alpha-7 nicotinic receptor as our model receptor. And we also decided to use a radio-labeled alpha-bungarotoxin as our labeled ligand. And alpha-bungarotoxin, as many of you probably know, is a great tool. So it binds to our human alpha-7 receptor in its orthosteric binding site, and it competes with small cholinergic ligands, which is super helpful for a competition type of assay. So I thought I would it might be helpful to talk through some common features of a competition binding assay, or one of our curves here. So this is a curve, a model curve. So this is just a calculated curve here. And once again, we are competing two ligands, are labeled and unlabeled, for a single binding site. And so for our purposes, at the far left-hand side of our curve, when we have very little unlabeled ligand in our reaction mixture, our receptor is half bound to alpha-bungarotoxin. And it's half bound because we use a concentration of toxin that is equal to its association equilibrium constant from the receptor. And as we increase our concentration of unlabeled ligand, our receptor becomes fully bound to unlabeled ligand. And now we fit our curves with a single component Hill equation, and we are essentially able to estimate two different parameter values from this equation. So our half competition concentration, which is equivalent to an IC50, as well as a Hill coefficient. And the Hill coefficient is essentially the measure of the steepness of your curve, but what it really helps to tell us is how our composition of receptor conformations changes in going from one end of our curve to the other, or as a function of our unlabeled ligand. Now, these two parameter values are essentially made up of all the individual binding and gating equilibrium constants, which can be a little bit complicated. So let's start simple. So for the purposes of display, we will ignore the desensitized state for now, but we know that we have a channel that can fluctuate between a closed and an open state. And we also know that our channel has a binding site. So our closed channel can become bound with ligand as well as our open channel. And once bound, our receptor can interconvert between the two states. And as all of you know, this interconversion is what we call gating. So this is a single thermodynamic cycle. And if we work to apply some probabilities to each of these steps, we know that our probability of our channel opening when unliganded is low as compared to that when bound with an agonist, for example. So by means of what we call the principle of detailed balance or the principle of microscopic reversibility, we know that our ligand binds our closed channel conformation with low affinity, and it binds to our open channel conformation with high affinity. So if we scale this up, still kind of pushing the desensitized state to the side just for simplicity purposes and clarity, we can see that if we look at our model receptor, which has 5 binding sites, and we have our 2 ligands competing for the same site, things can get really pretty complicated pretty quickly, and that it would be very hard to discern any of these individual equilibrium constants from our 2 observable values. So now that we had established our theoretical framework for our assay, I thought I would talk through a few of the key aspects that we went through that I found during our optimization process. So this includes the determination of our approach to equilibrium, as well as the importance of the characteristics of our labeled ligand, and then finally the importance of accounting for ligand depletion. So it probably sounds trivial, but 2 of the most important variables that we had to optimize in determining our approach to equilibrium were the time and the temperature of incubation. So first we started by incubating our reaction mixtures for 24 hours, and we wanted to see how our competition curves changed as a function of our temperature. So in going from 4 to 37 degrees. And we can see that as we increased our temperature, our competition curves not only shifted to the right, but we also saw that second component that we can see with our cyan curve disappear. And so, initially we thought, the second component could be an out of equilibrium type of artifact, especially because it disappears as a function of our temperature. But we also know that a curve with multiple components could be a naturally occurring phenomenon. So there could be some form of cooperativity between the binding sites, for example. And so, we had to take a step back and say, does our receptor behave differently at 4 degrees? And so what we did next was we ran our competition curves at 4 degrees Celsius, and we looked to see at how our curves changed as a function of our incubation duration. So we can see that as we increase our incubation duration, all of our labeled ligand becomes outcompeted by unlabeled ligand, and we lose that kind of second component that we are seeing in that cyan curve there. And so this helped to tell us that that second component was probably an out of equilibrium type of artifact. So at this point, we were leaning towards, okay, let's run our incubations at 37 degrees, but let's just double check to see how our incubation duration affects our curves at this temperature. And we can see that our curves are a lot tighter together. They bottom out much nicer than the curves in the previous panel. And any kind of slight second component that you might see with our curve at 4 hours is essentially eliminated as we increase our incubation duration. So at this point, we had established our incubation conditions, and we were deciding to run our assays at 37 degrees for 24 to 48 hours from this time forward. Now, the second key aspect that I wanted to talk about was the importance of the characteristics of the labeled ligand. So alpha-bungarotoxin is a wonderful tool. It's very easy to label because it's commercially available labeled, which was great. We were able to skip some of those initial steps. And it has a high specificity for our target receptor. So we could, feel a little bit easier about, any kind of off-target binding that might occur. And then finally, it dissociates slowly from the receptor. So we were able to perform extensive washing steps to further eliminate that non-specific binding. And finally, the third key aspect I want to talk about is the importance of accounting for ligand depletion. So at equilibrium, it's important that we know our concentrations of unbound ligands, so both labeled and unlabeled. And our labeled ligand, that's pretty easy. We can measure that, which is in the supernatant. But for our unlabeled ligand, that becomes a little bit more difficult. And so what we do is we assume that our concentration of unbound unlabeled ligand is equal to that of the total ligand that we add to our reaction mixture. But for this to hold true, we know that we need a limited number of receptors in our reaction mixture as to essentially make our ligand depletion negligible. But this is a little challenging because we also need a high enough signal to noise ratio, because we need to actually generate a competition curve and not just a flat line. So what we do for every individual competition curve that we run, we also measure our concentration of unbound labeled ligands, so our concentration of unbound alpha bungarotoxin. And we found that our amount of receptors in our reaction mixture was adequate, so our signal to noise ratio was adequate, when our concentration range of unbound alpha bungarotoxin was equal to its KD plus or minus 0.1 nanomolar. And these results were actually, really reproducible and very consistent. And it's something that I run for every single curve as a nice control. So now we had our methodology optimized. It took a while, but we were able to really hone in our reaction conditions. And so we wanted to ask how our observables, so how our half competition concentration and how our Hill coefficient relate to the underlying mechanism. And so what we did is we generated these calculated competition curves to better understand how our two observable values relate to our underlying mechanism. And what we did was we varied our different binding and gating equilibrium constants. So here what you see is our varied unliganded gating equilibrium constant. And we can get a better understanding of how our half competition concentration and how our Hill coefficient change in going from a receptor that is almost all the time in the low affinity state all the way through a receptor that is almost all of the time in a high affinity state. And so a lot of this stuff, wasn't always intuitive to us. So having these curves as kind of a framework was really helpful. And we also did this for our dissociation equilibrium constant from the closed state, as well as that from the open. Again, trying to get a better idea of how our observable values change as these internal equilibrium constants change. So with that, just a few final thoughts. We worked to optimize this methodology, but we're careful and paired it with a theoretical framework and these generated calculated competition curves to get a better idea of how our two observable value or parameter values change. And this will hopefully help us to gain mechanistic insight into function, especially when electrophysiological techniques aren't possible or aren't feasible for any kind of conditions that you may have. And with that, I would like to thank, of course, our funding agency, the NIH, Claudio, for all of his guidance with this project. I've been with him for a while. So I'm so thankful for everything that he's done, as well as our current and former lab members, Giselle and Pramod, for their support. And thank you so much for listening, and I'd be happy to take any questions.
Thank you, Nicolle. This was great, very clear, very logical, and very nice, very beautiful results. The presentation is open for discussion. Okay, Jean?
Nicolle, that was a wonderful talk. Thank you very much. So I'm intrigued by that observation that you reported that the time to reach equilibrium, or that second component of the binding curve that you're seeing, goes away with the elevated temperature. So are you attributing that totally to a temperature-dependent conformation of the receptor itself?
So we don't think it's temperature dependent. So essentially, this curve that you see,
where it goes away
here, right?
Yeah.
So this is right. We're varying temperature here, and so as we increase our temperature, we see it go away. So our initial thought was, hey, 4 degrees, maybe the receptor behaves differently. So then this is where I ran everything at 4 degrees, and I just increased my time. And so that cyan curve that you saw before is the exact same cyan curve. So if we just wait longer, we see that cyan curve flatten out. And so we, it was more of that out of equilibrium state that, at 4 degrees, things move slower. So we just had to wait longer. And obviously it's not super feasible to run assays at 96 hours, or else I think my PhD would have taken double the time. But yeah.
But is that, I guess what I'm saying is that an effect on the conformation of the receptor, and it's just being revealed? It takes a lot of time. Yeah.
I don't, that's a good question. I don't know. I think we attribute it to being slower, but things can change. I mean, it could behave differently at 4 degrees and 37, and it just kind of evens out, to the same thing that we're seeing.
Great, thank you.
John?
Yeah, I really appreciate just how thoroughly and carefully and theoretically and practically you've investigated your system. I had some question, just a general question about some of your modeling at the end. You have this really interesting system where you have not just 2 types of ligands, but you have 5 binding sites for each, as you mentioned, and you noticed some pretty profound changes in hill slope due to some kind of cooperativity there. Do you have, just based on the modeling you've done, do you have a kind of a more vernacular explanation for when you're seeing such profound changes? What's going on with those steepnesses that are even steeper than the regime that you see experimentally?
Right. So we actually assume that our 5 binding sites are identical and independent to one another. So that's how our model is constructed. So with our Hill coefficient and the range you can see down here. So yeah, and going from one end when our channel's super low affinity form to the other end when our channel would be in the very high affinity form for this particular model here, we expect a Hill coefficient of 1 because our composition of conformations isn't changing. One of the things that, so we see here probably goes up to about 3, and we don't see any values of 3 experimentally. I think experimentally, I think the highest Hill coefficient that I've seen is about like 2.0. And, for these particular calculated curves, we established for our other variables that we weren't changing, our dissociation equilibrium constant from the closed state is this particular concentration, from the open this particular concentration. And so we don't know how these Hill coefficient values would change if we changed our stable parameter values. And so that's absolutely something that Claudio and I can do and model something differently to see how much change we can see in the Hill coefficient. So yeah.
Very cool. Thank you.
Well, thank you. And Baron?
Hi, Nicolle. This was really fascinating to hear this presentation. I was wondering if there are any electrophysiological measurements of this receptor at 4 degrees and 37 degrees, because presumably it's all of what we know is at 22 degrees, right? So how does the data look, and what are the transitions look like? And is there any detailed modeling at those temperatures?
Yeah, that's a good question. And I mean, I know for us, it's not something that we've done here because we run everything at room temperature. And so I'm not super familiar. For these particular experiments, our goal is to use them when we can't use electrophysiology. So when we can't, run like we don't see any signal from our receptor. But I'm really not sure how this would relate to how we would see the electrophysiological results at 4 and 37, because we haven't run those here, and I'm not super familiar. But that's absolutely something I should look into to kind of compare what I'm seeing to what others are seeing with the other technique.
Yeah, I mean, circling back to Jean's question, there must be some of the transitions are happening at a much slower time scale. That's why the equilibration is taking much longer at lower temperatures.
Yeah. Absolutely. Yeah.
Great. Thank you.
Thank you.
Can thank Nicolle and all of our panelists today for fantastic talks. Thank you very much. And thank you for having an exciting set of discussions. And I think we can close the session.